Thank you for the invitation to speak today, and it does follow on nicely, hopefully, from Jackie's presentation. And it's good that I think the conference has achieved one thing already in getting some positive news, and hopefully one of the problems that I'll also present maybe moving forward a little. So that's good. My name is David Bunton. I'm from Biopta. We are a contract research company based in Glasgow. And we work with pharmaceutical companies to help them make better decisions about which drugs to take into clinical trials based on human safety and efficacy data at a preclinical, non-clinical stage. And what I want to talk about in the next 10 minutes is whether there is currently a place, or may become a greater place, for human tissue research in the whole process of drug approvals. So I wanted to start by presenting this graph, which is actually, it's about 8 years old now, but the numbers were reviewed last year and pretty much hold true. On the y-axis it shows, just a laser pointer here. This shows clinical failure rate of drugs that enter clinical trials, so the percentage attrition, and then the different reasons for that failure. You can see that in the 90s, in the sort of blue graph there, and then in the next decade, in 2000s, there were slightly different reasons. But the major ones highlighted, the arrows here: safety, toxicology, which we could broadly group together into adverse effects, and then lack of efficacy. In between them, they make up around about 70% of the total failures. So we really believe that using human tissue at the right stage, and we're fortunate in having Bob Coleman here, who is a pioneer in this area, in actually saying, look, the current approach doesn't work particularly well, and it seems obvious from this graph. There really should be more done to use human tissue to predict safety and efficacy. However, what I wanted to do next is just take a step back into the guidelines. These are the guidelines, which I'm not going to go through in detail. The ones that are most appropriate for testing the acute effects. So when we first go into clinical trials, looking at the short-term pharmacology of new drugs. And there, the main concern is, does it affect the central nervous system, the cardiovascular system, the respiratory system? These are the things that obviously are going to be most worrying in short-term exposures. So these S7A guidelines are international guidelines, and they do actually make mention of various parameters that can be measured through human tissues, as I'll show. And they actually mention that data from humans should be considered when available. However, in practice, this is actually pretty rare. So does the existing approach to measure these things, such as blood pressure, heart rate, and so on, in the cardiovascular system, and there are similar guidelines on central nervous system and respiratory, does it work, and is human data generated at an early enough stage? Well, we've analysed drugs that have failed from 2000 to 2010 in Europe, and the various classes of drugs that have failed. About 19%, the largest percentage here coming in cardiovascular, other than as a non-specific organ damage, which is then broken down here. So for the purposes of this presentation, I'm just going to focus on cardiovascular, but also because one of the areas that we've really focused on trying to improve. And I wanted to show a typical study. What do we actually mean by using human data early on? Well, we had a company come to us where they hadn't used human data. They got into phase 2 clinical studies. They had lots of observations of blood pressure problems in the patients, and they expressed surprise because it wasn't shown in the preclinical dog studies. So we compared in vitro these responses, and we saw the clinical effect in the human blood vessels, in vitro, ex vivo. So when we got those samples from the sources that Jackie mentioned, but completely absent in canine. So this is the kind of anecdotal evidence that we've been building up. Another example of that, a bit more visual, is to show the types of responses to serotonin, just a hormone that's present in the gut and the CNS and the cardiovascular system, and showing responses of blood vessels. This is contraction of small blood vessels in human and then in dog. Completely different. And this is, I'm not actually just bashing animal experiments here. This is just to show that there are cases where the prediction is insufficient if you rely solely on animal experiments. And this is not just from Biopta data. Igor Efimov in St. Louis published last year in cardiac models, looking at two different cardiovascular drugs in mouse hearts and then comparing that in actual functional human heart tissue, and again pretty much showed the opposite effect. So clearly there are problems. The question is, how do we actually go about trying to solve these? Well, to follow on from what Jackie was saying, if we can source the tissue, and this is a nice gruesome picture after our coffee of some skin, subcutaneous fat from cosmetic surgery. If this can be sourced very rapidly from surgery or from non-transplantable organs for research and used in an appropriate manner, here we've got our scientists dissecting some blood vessels and then perfusing a blood vessel with drugs to cause contraction or dilatation of the vessel. We can create data that's really translational to the clinic, and that's much of what Biopta is trying to do, is create data that actually tells us something about the likely clinical safety or efficacy of those compounds. So currently this is mainly done in a fairly rapid fashion. The procedures are very similar to that for transplantation, in that you're trying to keep the tissue in good condition and use it as rapidly as possible. I think a similar problem to the video earlier on with the projector. You might be able to make it out there. This is a small piece of beating ventricular muscle from a heart that's been donated to research that couldn't be placed for transplant. And so, a picture paints 1,000 words really in terms of the responses here. If you can add drugs directly to this tissue, it's able to tell you about cardiac indices that you can't model as effectively in animals in many cases. So what I wanted to do in the last 2 or 3 slides was try and make a case for how this would work in terms of the regulations. The point I'm at present is that this type of example we've got here is actually pretty rare in the regulations. This is an example where we tested a client's compounds for blood pressure effects. This is dilatation of blood vessels. In the normal healthy blood vessels, you see a good relaxation. In the presence of this client's compound, there was quite a severe inhibition of those normal mechanisms of control of the blood vessels. And this hadn't been detected in any of the in vivo rat experiments. And then, in a similar vein, this is one that 2 or 3 centres have looked at, where we're actually looking at a drug that was withdrawn because of an incidence of cardiovascular ischemia, which was deemed unacceptable by the regulators. We saw some effects in small coronary arteries that may indicate a potential mechanism for this type of side effect. And again, not apparent in animal models. So to finish off, I don't want you to attempt to take in all of this content here. What this is to show is that the existing models, the existing regulations do make mention of human tissue, but it's really a side matter. What we propose is that the regulations could include human data to a much greater extent, and that many of those assays are already available. So for predicting arrhythmia, there's a mixture of cell-based assays and tissue-based assays that can be used. To predict cardiac output changes and blood pressure, similarly, these types of tissues that I've shown between the beating heart and the isolated blood vessels are very important. But I don't want to overstate it and say that it can replace everything. There is still a need for an integrated assessment, and currently that still requires the in vivo situation. And that's really where we round it off, because in most cases the drugs are studied in 2 animal species according to the regulations, and until we actually see a better access to tissue and a more really a greater evidence base from the type of research that we've heard earlier on, I think that we may have to wait a few more years yet, but certainly things are gathering pace, and end on a positive note, hopefully the conference today will help accelerate that. So thanks very much, and look forward to questions later.